So we're going to go through how to fill out this coloring page that you either have in your notes packet or I gave you separately. So first thing we have to do is we have to color each of the different items. So I'm going to go through the different items here. So DNA, pick a color, any color. If you don't have colors, you can just label it. DNA is double-stranded. So our DNA is going to be this whole thing here. So again, if you have colors, you can go ahead and color it a color. You can put the color here, or you can just label it. So DNA is right here. We have a nucleus. This is our nucleus. So the nucleus we usually draw as a circle, but this is part of it, this dark gray. So this is going to be our nucleus. And remember, the nucleus has a membrane around it. So that's what we're seeing here. We have ribosome. The ribosome is where translation's going to happen, where our proteins are made. That's this big kind of hamburger bun shaped object. It's actually made of two parts, which is why you see a big part here and a little part here. Our mRNA, or our messenger RNA, is being made from our DNA. So that's this guy right here. So messenger RNA and messenger RNA. tRNA, these guys look kind of like T's. They look like this. So the tRNA is going to bring in our amino acids. So these are tRNA. And they're carrying the amino acid puzzle pieces on the top. So these are amino acids. So it also says fill in the missing bases on the DNA, mRNA, and tRNA. Okay, so we have our DNA here, and we're going to fill in the matching letters on the DNA. So A pairs with T, A with T, C with G. So you're going to fill in a G, a T, and so on. Now, let's imagine we have a T up here. That's going to match with a letter for our messenger RNA. T always pairs with A, so we're going to put an A on this side for our messenger RNA. In the case of C here, C goes with G, so we'd put a G here, and then our mRNA is going to be C. In the case of a T here, T pairs with A, so we're going to put an A there. And then on this side, messenger RNA does not have T's, it has U's. So we're going to go ahead and we're going to put a U instead of a T. So you're going to go ahead and fill in all of those letters. You can copy those letters. So let's say I had A-T. That was my order. I could just write A-T. Just copy them all. This is the same messenger RNA here. It's just showing it moving over here. So you just copy these letters over here. And then our tRNA, we've got to match up letters again. And remember, tRNA is RNA. So we cannot use T's. So if I have AAA on my messenger RNA, I'm going to have UUU on my tRNA. Or if I have GGG here, I'm going to have CCC here. The last thing we have to do is label a codon, anticodon, and peptide bond. So codon, remember, 3 letters in messenger RNA that code for a protein. So I'm just going to circle 3 letters, or 3 letters here, or 3 letters here. So 3 of these DNA bases, that's a codon. I'm going to circle an anticodon that matches up with the codon. So anticodon's on the tRNA. It's 3 letters on the tRNA. And then a peptide bond is the bond here in between the amino acids. So that should give you everything you need in order to label your diagram.